Hello, I'm Dr. Mirko Pegoraro, and this is a lesson related to your workshop on sea urchin. In this lesson, we are going to talk about external fertilization. External fertilization occurs when the gametes, eggs and sperm, are released outside the body of the animal, and they meet each other outside the body of the animal. This is common in many aquatic animals, as you can imagine. So at the end of this lesson, you should be able to describe the processes of external fertilization and how they are similar or different compared to the process of internal fertilization. And you should be able to talk about the early stage of development of sea urchin. In fact, the animal that we are going to talk about to describe external fertilization is indeed sea urchin. So if you remember, when we talk about maturation of the egg in mammals, in human, we say that fertilization occurs in mammalian egg when they are at this stage of the meiosis. So when they are in second metaphasis. We indeed say that they only complete meiosis when they are fertilized. This is not the case for sea urchin species. They release in seawater eggs that already completed meiosis. In their eggs, when they are released in water, we can recognize indeed the female pronucleus surrounded by the egg membrane. So the sea urchin egg is, structurally speaking, quite similar to a human egg. We have similar structures surrounding the egg itself. One particular very important structure is called the vitelline envelope. The vitelline envelope is mostly made of protein and is very similar to, if you want, the zona pellucida that surround the mammalian egg. Here you can see the vitelline envelope on top of the membrane of the egg. One of the jobs of the vitelline envelope is to recognize, if you want, the sperm. So it is involved in the sperm-egg recognition, as well as protecting the egg. So the job of recognizing the sperm and the correct sperm is particularly important for animals like sea urchin that release their eggs in seawater. So they have external fertilization. This is because many different species of sea urchin may spawn at the same time, releasing eggs and sperms in the water. So each egg wants to recognize the correct sperm, so the sperm that belong to the same species. The egg also is surrounded by egg jelly. The egg jelly also is involved in attraction and activation of the sperms. And if you want, the egg jelly is similar or does a similar job to what the cumulus does in mammalian eggs. In the sea urchin egg, we also recognize a layer just below the membrane. So here is the membrane with the villi. Just below the membrane, we recognize a layer that is called the cortex. And in the cortex, we have the cortical granules. The cortical granules of the sea urchin egg are very similar to the cortical granules that we saw already in the mammalian egg. Excuse me. So, the process of recognition between the sperm and the egg are very similar between sea urchin, for instance, on the left, and mammals on the right. We have a chemoattraction initially, so the sperm is chemically attracted to the correct egg, so they swim toward the correct egg. Then we have a process of exocytosis, which we don't see in mammalian sperms. In the process of exocytosis, we have the production of what is called an acrosomal process. So there is a protrusion out of the head of the sperm. And this happens when we have the acrosomal reaction. Okay? The acrosomal reaction happens around the jelly layer. Similarly, the acrosomal reaction was happening around the the cumulus in our in the mammalian egg. The enzymes that are released from the chromosomal after the chromosomal reaction, so when the acrosome is open, so that those enzymes allow the digestion of part of the jelly layer, so the sperm can swim through the jelly layer. So this is called the passage. Finally, we have binding of the sperm initially to the vitelline envelope, and after the binding to the vitelline envelope, the membrane of the sperm and the membrane of the egg can fuse together. This allows the passage of the genetic material from the sperm to the egg. So these final steps are very similar in the sea urchin, for instance, egg and sperms, and in the mammalian egg and sperms. Obviously, one big difference between mammals and sea urchin and other aquatic animals is the production of sperms and eggs. Indeed, when they spawn, there is a large production for sea urchin of both sperms and eggs. Very interestingly, the sperms inside the testes of a sea urchin are immotile, so they cannot move, and they become motile when they are released in seawater. And the reason why they change from being immotile and motile is the pH of the testes compared to the seawater. Indeed, the pH of the testes are a bit more acidic, so pH is 7.2 compared to seawater, which is a bit more basic, pH 7.6. Why do I mention this? I think it is important to mention this in relation to climate change. So you see, one of the consequences of climate change is the acidification of seawater. So if this seawater, which has a pH of 7.6 usually, become more acidic, so become more like the testes, then it is possible that the sperms do not become motile in even when released in seawater. So it becomes more difficult for sea urchin and other aquatic animals to actually reproduce. So it is extremely important, as I mentioned before, for sea urchin to be able to attract and activate the correct sperms. There may be different species of sperms around, so this particular egg wants to activate and attract only the sperm that belong to the same species, not others. And one way that sea urchin species make sure that they activate and attract only the correct sperms are via these peptides called sperm activating peptides, or SAPs. These are obviously species specific, they are different for each species, and indeed one of these SAPs is called RISACT, and this belongs to this particular species of sea urchin, Arbacia puntuatua. So if you look on this picture, you see the effect of RISACT on this species sperms. So at the beginning, this picture with 0, so at the time of adding the RISACT, you see that all the sperms are swimming in circle. They don't have a direction, they don't know where to go, they don't swim too fast or too hard. However, immediately after adding the RISACT, the sperms start to swim more strongly, more quickly, and toward the position of where we add the RISACT. And you can see how the sperms move to that position when you take picture at different time point. Now, we mentioned already when we were talking about the mammalian egg and during the fertilization that it is important to block polyspermy, so to avoid polyspermy. So to avoid that multiple sperms fertilize the same egg. It is even more important for sea urchin since a single egg can be surrounded by thousands of sperms and sperms of different species too. We recognize in the sea urchin egg and during the process of fertilization two kind of block of polyspermy. One is called fast block of polyspermy and one is called a slow block of polyspermy. So the fast block of polyspermy is called like that because it happens very fast after fertilization and the duration is quite short. The fast block of polyspermy is related to a change of the electrical potential of the egg membrane. So you can see here in this picture that the normal resting stage membrane potential for the sea urchin membrane is negative, minus 17 millivolt. So it is, but what is important to remember is that it is negative. Soon after fertilization, so when the sperm fusion occurs, this membrane potential change and become positive, plus 20 millivolt. And you can see here the change become positive. Now, sperms cannot bind to membranes that are positively charged. Okay, so in this way, with this change in the membrane potential after fusion, other sperms cannot bind to the membrane at all. But this change of membrane potential only lasts for a minute. And after a minute, as you can see here, the membrane potential already become negative again. This initial fast block of polyspermy allowed the time for the slow block of polyspermy to happen. And the slow block of polyspermy is basically a cortical granule reaction. We already saw that happening in the mammalian, during the mammalian fertilization, where the granule were opening and releasing their content and digesting basically the zona pellucida. And here, similarly, the content of the granules are released and they detach, their enzyme that detach the vitamin envelope from the surface of the egg membrane. Okay, so if you look on this picture, you see that fertilization occurs here where the red arrow is, and then the membrane, the vitelline envelope, get digested and released from the, or if you want, cut loose from the membrane of the egg. But this structure, so the vitelline envelope, do not really go away. It just get cut off the membrane, and so it become separated from it, but stay in place, forming what is called a fertilization envelope. So now the fertilization envelope will block any of the sperm that try to enter or to fuse again. It is also detached from the egg membrane, so even if some of the sperm were able to pass through the fertilization envelope, it would not be able to actually fuse to the egg membrane. Also, because it's made of protein, the fertilization envelope, mostly protein, it functions as a protection for the first stage of development of the sea urchin embryo. Okay, so now we have a fertilized egg, we have the formation of a zygote, and as in humans, as for the human embryo, the first stage of development include cleavage and gastrulation. So let's have a look on the first stage of development for sea urchin. So we had external fertilization. Now we have a look very simply the first stage of cleavage and gastrulation. The cleavage in sea urchin is called radial holoblastic cleavage. It has similarities and also differences compared to our cleavage. So first of all, the first and second cell division, the first and second cleavage, are both meridional in sea urchin embryo and perpendicular to each other. So if this is the zygote, the first cell division is meridional from top to bottom and produces two left and right cells. Then the second cell division is perpendicular to the first one and is also meridional and produces these four nice looking cells. Now the third cell division is now equatorial and produces two groups of cells, four on the top, that are part of the animal half of the animal pole, and four on the bottom, which are the cells of the vegetal pole or the vegetal half. Now the fourth cell division is quite peculiar in this animal. It is peculiar because the cell on the top, so in the animal half of the cell, these four cells here, divide meridionally from top to bottom, all of them, and they produce cells that are quite similar in size, called mesomers. However, the cells on the bottom, the cells on the vegetal earth divide equatorially, but they don't divide in the middle. In fact, they produce 4 cells that are quite large with most of the cytoplasm, called the macromeres, and 4 cells here on the bottom, which are quite small with little cytoplasm, called the micromeres. Okay, so mesomere on top, 8 cells, all similar in size, then 4 macromeres with most of the cytoplasm, very big, and then 4 small micromeres on the bottom with a small amount of cytoplasm. Now, this is a vision of the sea urchin from the bottom after another stage of cell division. And it is interesting because, again, the mesomeres divide meridionally and produce cells of the same kind of size. So on top there you see the mesomere of a similar size. Then the macromere divided in 2, but equatorially, but again producing 2 groups of cells of different size, veg 1 and veg 2. Veg 1 tend to be a bit bigger than veg 2. And the micromeres divide again equatorially, but not equally, producing very small cell called the small micromeres, and somehow a bit larger cell called the large micromeres. Okay, so the blastula at this point, like our blastula, start to produce by cavitation a blastocoel. And you can see here in these 3 pictures of the sea urchin embryo at different stages of development, the position of the blastocoel, and now the blastocoel become bigger and bigger. This picture is quite nice because you can see actually the micromeres here, and on top there you have the macromeres. Now by the 9th or 10th cleavage, the cells all around the blastocoel start to produce cilia that point outside, okay, not inside the blastocoel, but outside. And then the vegetal pole, the cells of the vegetal pole become a bit thicker, they thicken a bit, and they form what is called the vegetal plate. Here you are, so you see here this thickened group of cells that form the vegetal plate. Now, similarly to what's happened to our own embryo, the animal pole cells start to produce enzyme that digest the fertilization envelope, so we have an arched blastula. Remember, at one point, even our embryo need to digest a hole out of the zona pellucida and to arch out the zona pellucida. So it is very similar of what's happening here with the arched blastula. And in this cartoon, if you want to represent this arch blastula, so you have here an arch blastula with all the cilia all around. You have on the bottom here, recognize the vegetal pole, and on top the animal pole, and here the region that is more thick is called the vegetal plate. Now, the central cell of the vegetal plate start to migrate inside the blastocoel, and now we are already in the process of gastrulation. So we are moving the cell around. Okay, so these cells that move inside the blastocoel are called the primary mesenchyme. These primary mesenchyme cells are the ones that become the future mesoderm of this animal. The cell all around here will become the ectoderm, so the blue cells all around the blastocoel, and the rest of the cell of the vegetal plate, here indicated in orangey yellowish, will become the future endoderm. So at one point, the cells, the region of the vegetal pole, the region of the vegetal plate actually, started to move or invaginate and form a depression. This depression, this opening, if you want, is called a blastopore. Okay, so this depression that is forming is called initially a blastopore. Now, here you have that the blastopore, thanks to filipodia, get elongated. So these filipodia are forming from the top cells of the blastopore. So these are the initial the vegetal plate cells. From this filipodia, they attach to the blastoquel surface, and they push up the depression. They elongate and enlarge the depression here of the blastopore. In this way, forming what is called the primitive gut. You can see here the primitive gut, also called archenteron. So the archenteron is the primitive gut that is formed by this filopodia, pushing up, taking up, moving up the initial depression formed here that identify the blastopore. So here you still have this opening that is called the blastopore. And here this is a cartoon representation, okay, where you have, this is the primitive gut, also called the archenteron. The filipodia already make this growing quite a lot, and at one point the archenteron, the upper part of the archenteron, will contact the blastocoel surface. And when this happens, the blastocoel and the archenteron fuse together and create an opening. This opening is going to be the future mouth. Okay? The future mouth. And the blastopore now will become the future anus. So the blastopore is here, this opening here, and will become the future anus of the animal. So at this point, the embryo will go through a process of metamorphosis, and the embryo form this Pluteus larva. Okay, this is like a triangle or star shape, Pluteus larva. Okay, and what you can see in the Pluteus larva quite clearly, apart from the cilia, you can see the anus opening, and this anus opening is this anus opening in this picture here, which basically is the blastopore, the original blastopore. And the mouth opening here, formed by the archenteron binding to the blastocoel or fusing the blastocoel, is now forming the oral cavity of the Pluteus larva. Now, during the workshop exercise, you will have to use the information in this lesson plus the information that you can find in other websites that I indicated in the guide, in the workshop guide. You will need to identify in pictures that I give you in that particular guide different parts of the sea urchin larva at different stage of development. For example, you will need to identify macro and micromeres, the blastocoel, perhaps the animal and vegetal poles. You will also need to identify the blastopore, the archenteron, and the filipodia, excuse me, as well as the position of the mouth and the anus in Pluteus larva, for instance. So, at the end of this lesson, at the end of the workshop exercise, you should be confident in describing external fertilization in sea urchin, and as well as initial developmental stages of sea urchin embryo.